Thank you, everybody, for attending the Early Age Onset Colorectal Cancer Disparities Society Meets Biology session. It's really my privilege to have organized and shared this session with Dr. Perea. And I'd like to start by thanking Fight Colorectal Cancer for recognizing the importance of this growing topic of disparities within the early onset colorectal cancer population. And so without trying to take time away from our speakers and jump right into the science, I'd really just like to kind of set the stage to highlight the importance of early age onset colorectal cancer disparities and save most of my remarks for the path forward section after each of our phenomenal speakers. So I'd like to just give you a couple different facts overall that can stand on their own, but together make quite an impact. So it's known in the overall population that colorectal cancer incidence rates are nearly 1/3 higher among men, yet women have a higher incidence of right-sided colon tumors. Specifically within the population of early age onset colorectal cancer patients, however, the proportion of right-sided colon tumors also differs by age. Adding to this complexity, men harbor a 12 to 18% increased hazard of disease-specific death among early onset colorectal cancer cases, yet regions of disproportionately high mortality from early onset colorectal cancer not only differ by sex, but are largely concentrated in the southern U.S. And among women, physical inactivity is a community health behavior that modestly correlates with hotspot residence. Evidence is emerging, and you'll hear more upon today, that metabolic phenotypes of colorectal cancer differ by sex biologically. And kind of shifting transitions to a bit more on the demographics, we know that the proportion of individuals with early age onset colorectal cancer is nearly twofold higher among people of color. Yet the proportion of Hispanic cases diagnosed with early onset disease increases across younger age groups. Biologically, recent study in JNCI demonstrated differences exist in epigenetic aging of normal colon by race, and my group presented the tumor mutational burden varies by race in early onset colorectal cancer. I presented to you about 10 different facts that each alone can stand to serve as a talk in a session or comprise an entire session to discuss, but together they accumulate to drive one large point and impact, and that's that there persists a disproportionate burden of early onset colorectal cancer across diverse population subgroups, one that we don't fully understand yet and need to put energy towards elucidating. And I hope that today's session will help shed light on where we've gone thus far and where we have yet to go moving forward. So it's really my privilege to have three amazing colleagues and speakers comprise this session today. You'll hear from Dr. Stoffel, who's an associate professor of internal medicine at the University of Michigan and director of the University of Michigan Cancer Genetics Clinic, where her interests encompass early detection and prevention of gastrointestinal cancers. From there, we'll jump into Dr. Johnson, who's an assistant professor at the Yale School of Public Health, whose research aims to understand sex-related differences in colorectal cancer metabolism related to environmental exposures. And we'll round out with Dr. Suet-Yi Luong, who is a professor in natural sciences and chair of gastrointestinal cancer genetics and genomics at the University of Hong Kong, with interests focused on the molecular genetics and genomics of gastric and colorectal cancers. And I hope throughout these different speakers, you'll really begin to encompass the topic of early onset colorectal cancer disparities from society all the way through biology. And with that, I will let Dr. Stoffel go ahead.
Well, thank you very much. It's such a pleasure to be this really important symposium. And I think that one of the things we can hopefully all do together as a result of this meeting is to be able to move the path forward. And there's so much we have to learn and so much we can do together. So by way of introduction, as everyone knows, colon cancer risk assessment relies really heavily on age, family history of colon cancer in first-degree relatives, and a personal history, and knowing the personal history of colorectal adenomas or inflammatory bowel disease. in my role as director of the Cancer Genetics Clinical Program at University of Michigan, my focus is on genetics and what can we learn from young individuals with colon cancer with regard to genetic predisposition. Well, this slide of this 39-year-old woman who presented with colon cancer at age 33 is a perfect example of everything that has been discussed during this forum over these past 2 days. Had somebody collected a family history that had identified that her mother had also developed colon cancer at the age of 45, as well as her grandfather and multiple other relatives, perhaps somebody would have put 2 and 2 together and figured out that this family history is really classic for Lynch syndrome, which as has been discussed, is the most common hereditary colon cancer syndrome. And we know now that the spectrum of hereditary predisposition to colorectal cancer goes beyond Lynch syndrome, and in fact goes beyond some of the genes typically associated with colorectal cancer. This figure, we developed this figure to just show the overlap between the different common hereditary cancer syndromes, which include not only the hereditary gastrointestinal cancers, but also genes associated with genetic predisposition to breast cancer and prostate cancer. And importantly, the prevalence of Lynch syndrome is in the general population is about 1 in 300, which is the same as the prevalence of hereditary breast ovarian cancer. However, most patients have heard about BRCA, but very few people have heard about Lynch syndrome. I think looking at the prevalence of germline mutations in colorectal cancer patients by age group, we know that approximately, that among individuals diagnosed with colorectal cancer over the age of 50, approximately 1 in 10 has a pathogenic germline variant. But among individuals diagnosed under the age of 50, it's 1 in 5. And the younger you get, the more likely it is that there is a genetic predisposition. These data here are from our group at University of Michigan. I'm also presenting data from the MD Anderson group that specifically looked at the prevalence of germline mutations among individuals diagnosed under the age of 35, which was 30%, as well as the prevalence of pathogenic germline variants in the Ohio population presented by Rachel Perlman and Heather Hampel, which found an overall rate of 16%. So in all 3 of these studies, the prevalence of germline mutations is fairly similar, and the germline mutations identified, as mentioned, go beyond Lynch syndrome and include genes associated with other conditions. So, but we know that most patients who present with colorectal cancer at a young age have family histories that are less remarkable than the one I just showed you. And in fact, what if this person had presented? What is the likelihood that she would have had germline genetic testing? Well, I would say that probably 5 years ago, we would have said the likelihood of a germline mutation is very low because this person does not have a family history of colon cancer. And this person, lo and behold, has a pathogenic germline variant in PMS2, which we now know has variable penetrance. This person could very easily also have an APC germline mutation associated with familial adenomatous polyposis, because what we're seeing now is we're seeing that some individuals are presenting with less striking polyposis phenotypes and more attenuated polyposis presentation. Likewise, this patient could very well have Li-Fraumeni syndrome with pathogenic variants in P53. But overall, it's important to note that 4 out of 5 young people with colorectal cancer have no pathogenic germline variant identified on their genetic testing. So what does this mean? When we looked at our cohort accrued through University of Michigan of 430 young individuals with colorectal cancer, we see that there are differences between the folks who have a positive genetic test and no mutation identified. Specifically, only 25%, only 1 in 4 young people with colon cancer has a family history of colon cancer in a first-degree relative. But importantly, even among mutation carriers, in our cohort, only half had a family history of colon cancer. Unfortunately, individuals who don't have germline variants and don't have a family history are most often diagnosed at later stages for exactly what everybody's been discussing, which is the tendency to say, no, you're too young to have colon cancer. Those symptoms may not need to be evaluated. And we now know that that's not true. And as a gastroenterologist, I actually teach all of my GI fellows and all of the primary care doctors that I work with that we cannot disregard symptoms in young people. And certainly the prevalence of rectal cancers is much higher in individuals who do not have pathogenic germline variants. So where are these cancers coming from? Well, for a while, we had thought there was the missing genetic contribution for complex diseases. And this figure, published first by Terry Manolio and colleagues, shows us that there are some rare, strongly penetrant genes, as we mentioned previously, that are associated with big effect sizes in terms of colorectal cancer risk. And we also know that there has been work identifying smaller single nucleotide polymorphisms through GWAS studies, which may have a smaller effect on colorectal cancer risk. However, we also know that it is most likely that there is something else going on other than genetics. And I think that Caitlin Murphy's study that demonstrated the birth cohort effect is probably the most striking evidence to support this, in that in that the curve sharply swerves upward among individuals born after 1960, and particularly that millennials have a colorectal cancer risk that's approximately 4 times higher than their grandparents. And I think that why is this? Well, it could be because of multiple things: diet, microbiome, antibiotic use by humans, antibiotic use in livestock and poultry. And I think that where we're going now is looking more at the environmental factors. And when we look at the disparities, looking at the age standardization rates for colon rectal cancers across the world, it's really important to see that the rates are highest in Europe, Australia, and the Americas. But when we look at the United States in particular, we can see a very striking disparities even among even in the map of the United States, with the darker red areas constituting the areas with the highest incidence of young onset colon cancer. As has been mentioned previously, if you superimpose this with the map of where we see the prevalence of obesity, there's remarkable overlap. But we also know that there are many young people who develop colon cancer who are not obese and are not sedentary. And so we need to move beyond this. Looking at the incidence of early onset colorectal cancer by race ethnicity, the purple curve at the top represents African Americans. The green bars that are going up are white, non-Hispanic, and the red represent Hispanics. And as we can see, the curves are not equal for all of these different ethnic groups. So what does this tell us? Well, in terms of racial disparities in colon cancer, as Dr. Holowati just mentioned, colorectal cancer incidence and mortality are highest among non-Hispanic Blacks, and the incidence of colorectal cancer under the age of 50 is 2-fold higher for non-Hispanic Blacks. We know that there are racial disparities in colorectal outcomes, especially among individuals with advanced stage diseases. And is this indicative of a lower uptake of screening, differences in cancer treatment, or differences in tumor biology? Well, I had the pleasure of working with Dr. Holowati a few years ago on a study that compared survival among individuals with young onset colorectal cancer by race. And this SEER analysis, these curves here, show us a very interesting picture in that African Americans, represented by the blue line, have worse survival at every stage disease, but the survival disparities are greatest among stage 2 disease. And what's interesting about this is that young Black patients with stage 2 colorectal cancer are 60% more likely to die of their disease compared to their young white counterparts. Is this differences in tumor biology? Is this differences in cancer treatment? Or is it interaction between the two? And this figure that Dr. Holowati has published, I love, because this basically shows us that we've focused on biology and genetics, but there is a lot more out there that we need to be able to look at to identify what individual level factors in terms of health behaviors, what environmental factors in terms of stressors, neighborhoods, access to high quality food, and what systems are contributing to these disparities. And what determines whether a person receives high quality care? Patient's background, patient's attitudes, knowledge, self-efficacy, the information that their providers have, and also the system constraints, the availability of high quality care in their area. And this is where we can really make a difference. So in terms of looking at the modifiers of colorectal cancer risk, there's biology, there's environment, there's health behaviors, and there's some very exciting work going on looking at how can we improve screening, what is it about diet that makes a difference, how does the microbiome and inflammation interact to impact risk. So where do we go from here? Well, I think that one of the summaries of these two days that we've been together have been that it's so important to integrate colorectal cancer risk assessment into our routine health maintenance for all adults. All adults, regardless of age, should be screened for symptoms, not just individuals over 45 and over. But we also need to assess family history for everyone, starting in early adulthood, to look at family history of colon cancer, colon polyps, and other cancers as well, and to make sure that individuals who meet criteria for genetic evaluation have the opportunity to get that diagnosis and intervene early before a cancer develops. Likewise, as you'll hear from my colleagues in this session, studying tumors to identify whether there are associations with particular behaviors, diets, or exposures that may inform treatment and prevention, particularly with regard to targeted therapies for cancer, but also to be able to look at epidemiological factors and how these impact the biology of tumors. And finally, working to eliminate disparities in healthcare delivery is something that we can all move towards. Improving access to colon cancer screening and genetic evaluation, and standardizing colorectal cancer treatments. So on that note, I would like to hand this off to my colleagues in this session, and thank you so much, and we look forward to the breakout sessions afterwards.
Thank you, Elaine. And I'll transition it now to Carol.
Hi, I'm just about to share my screen. Can you hear me okay? Great. Okay. Can you see my slides? Oh, come on, okay, great. Okay, well, I'd first like to thank the organisers of this meeting for inviting me to give a talk in this important session. The aim of my talk really is to show you the importance of understanding sex differences in colorectal cancer metabolism and how it associates with prognosis for the patient. So much of my research so far has actually been conducted on biological samples from later onset colorectal cancer patients, but the technology that I use can evaluate both genetic and environmental contributors. Thus, I think it's really primed for examining early age onset colorectal cancer. So what's in most cancer types that are not in a reproductive organ, actually males have a higher incidence and mortality, and overall males have a higher incidence and mortality of colorectal cancer. But what's interesting in terms of colorectal cancer is that females actually have a higher incidence of right-sided colorectal cancer. So that's a primary tumour that occurs between the cecum and the hepatic flexure. Whereas sex differences in incidence are not really seen in left-sided tumours that occur between the splenic flexure and the sigmoid colon. And males tend to have a higher incidence of rectal cancers as well. So this is important to note, as patients with right-sided colorectal cancer have poorer clinical outcomes. So therefore, knowledge of where the lesion occurs in CRC is important, as there is a correlation to survival. So there are some molecular features that
Oops, I think we've shortly lost Caroline. So maybe in her absence, Dr. Leung, do you want to get started with your talk, and then we can have Caroline jump in on the back end?
Oh, Dr. Caroline is back. Maybe I think she can continue.
I'm sorry, did you?
You're just
I'm sorry, I think I might have lost internet.
That's all right. Go ahead.
My apologies. I'll just continue. I seem to have lost internet for a second there. So what we've seen in my lab actually is that the non-genetic factors that predominate on the right side of the colon are very interesting. So we've seen that there's genotoxic dietary metabolites, such as bile acids, which are increased in individuals on a high-fat diet, and are higher on the right compared to the left. And there's also differences in abundance between males and females by age, and they appear to affect immune responses. And we've also seen that right-sided colon cancers present with bacterial biofilms, which cause a pro-inflammatory state in the colon and also affect tumor metabolism. However, what this figure does not show us what the sex disparities are in early age onset CRC. So I hope to show you in this presentation that sex differences in CRC biology are important to understanding CRC risk, incidence, and outcomes, and thus must be considered in early onset colorectal cancer. As I mentioned, my lab is interested in identifying how metabolites could affect CRC incidence and outcomes. And metabolites are very important as they are derived from many sources, including the diet, and are required for building new tumor cells and providing energy. So metabolite levels also change in response to the nutrient supply and the microenvironment, where the tumor resides in the colon, to enable tumor cells to grow under different conditions and different stresses. And so this is sometimes referred to as metabolic rewiring. Metabolites can also inform us about any changes to overall metabolism through examination of metabolic pathways, wherein they're consumed and produced and regulated by proteins that are encoded by genes. In addition to the host regulatory processes of metabolites, the microbiome is also involved in co-metabolism of these metabolites and can generate their own metabolites that in some cases have been linked to CRC, such as colobactin. So in terms of sex differences in metabolite levels and metabolic pathways, we know that it's highly plausible that females and males can have a unique biology that's shaped by multiple influences. So for example, sex-steroid hormones and hormone receptors can regulate important signaling pathways in CRC. And males and females have different lifestyles, such as different types of diets. Females have pregnancy and breastfeeding, which can change the metabolic profile permanently. They may take different medications, so females may choose to take oral contraceptives or hormone replacement therapies. And in addition, microbial diversity and function likely differs between males and females. And then life course exposures may also be different. Therefore, males and females may have a very unique metabolic phenotype, which may associate with the tumor growth and with patient prognosis. So before I move on to any data, I'd really like to first highlight the tool that I use in my research, as I think it will be really valuable for early age onset colorectal cancer studies. So to measure metabolites and to examine their relationship in metabolic pathways, I use a tool called metabolomics. And this is the unbiased assessment of all the metabolites that could be in a tumor tissue or in another biological sample. So metabolites reside in various chemical classes. You may have heard of amino acids and vitamins and sugars and lipids. And really, metabolomics is a systems level tool similar to genomics or transcriptomics in that we measure thousands of variables in an unbiased manner. But what makes this tool really unique, and I think primed for early age onset colorectal cancer studies, is a simultaneous detection of metabolites that are derived from environmental exposures. So in fact, really, I think it's the only omic tool which can really allow you to evaluate both genetic and environmental influences. So as you can see from this diagram, each person has their own unique influences on the metabolome or on the metabolic phenotype or metabotype. So there are genome-derived factors, exposure from different environmental chemicals and diet, exposure that come from lifestyle choices such as smoking and alcohol use. Therefore, using metabolomics, we can really get a better understanding of these exposure-response relationships, disease heterogeneity, and also why some people do not respond to treatments. So therefore, early age onset colorectal cancer research, I think, would really benefit from the use of metabolomics to uncover these different environmental factors and biological changes that could be linked to etiology and prognosis. So the study I'm presenting to you now is a metabolomics assessment of tumor tissues taken from CRC patients that are aged over 55. But I'm hoping that this will demonstrate that male and female patients do have distinct tumor metabolism that associates with patient prognosis and needs to be studied further. So in this study, I collaborated with Dr. Philip Patey from Memorial Sloan Kettering Cancer Center and Dr. Khan from the Yale Cancer Center. Dr. Katie had collected tumour tissues from 750 cases in the 1990s. And what is special about this cohort is that the tumours were collected in the operating room and directly frozen in liquid nitrogen, providing a tumour source, a tissue source that I can use for metabolomics. So we cannot use any fixed tissues. Also, follow-up information was recorded, so we can link metabolites to prognosis. So we stratified the cohort by sex, stage, and anatomical location of the primary tumour. And I'd just like to point out two postdocs at the time that worked on this project that now work in different groups. So Dr. Rattray has his own group at University of Strathclyde, and Ping Tsai has now moved on to Chinese Academy of Sciences, and they really drove this research forward. So to analyze the tumours, we took 10 milligram of tissue, we homogenize it, and we extract out the metabolites, and then we inject them onto our liquid chromatography mass spectrometry system. And here is a diagram of it here. Basically, this instrument can isolate, ionize, and weigh each of the components that are in the tumour homogenate. And we can get a relative concentration of each of the metabolites within the sample. We end up with a very high dimensional data set of about 15,000 variables, but we have a bioinformatics workflow that we can do batch alignment, normalization, and statistical analysis to pull out those important metabolites. So overall, we saw a really stark difference in tumour metabolism between males and females. There was one metabolite and metabolic pathway I'd really like to focus on, which is asparagine. So asparagine can come from external sources, such as a diet. It can be metabolized by the microbiome, and it can be produced intracellularly via an ATP and glutamine-dependent reaction, which is catalyzed by asparagine synthetase, or ASNS, as I showed in the diagram below. And this reaction and gene has actually been previously associated with poor prognosis for patients with different types of brain cancers. So what we have here are 4 subgroups of patients, females and males with right and left-sided colon cancer. And we have the abundance of asparagine and glutamine in each of these groups. And you can see that for, we have normal colon tissue, and we have stage 1 to 3 tissues. And for females with right-sided colorectal cancer, significantly increased amount of asparagine and glutamine in their tumor tissues, where we do not see this in the male patients or in the left-sided patients. There was a previous publication back in 2016 as well, which noted that asparagine production could be linked to nutrient depletion of the tumor and the uptake of additional, which could drive the uptake of additional amino acids, in particular threonine and serine, fuel cancer growth and control endothelin, sorry. Therefore, we examined whether the amino acids could correlate with increases seen in asparagine. And you can see from these graphs where we have asparagine plotted against these amino acids, excuse me, that there's a significant between all stages, but the same effect is not seen in males. In tandem, we sought to validate our work using transcriptomics data from these publicly available data sources. And we examined differences in mRNA expression between females and males with right and left-sided colon cancer. And we saw again that these mTOR pathways were enriched in females with right-sided colon cancer. So we then sought to provide clinical relevance. So we investigated if asparagine levels associated with patient prognosis. This talented MPH student in my lab, Ginny Shen, used multivariate Cox proportional hazard regression models to examine 5-year overall survival and recurrence-free survival associated with asparagine stratified by sex. So you have 2 models here, model 1 and 2. In model 1, asparagine was treated as a continuous variable, and we saw that it associated with both shorter overall survival and recurrence-free survival in female patients in CRC. And in males, we saw that this was a negative association. And in model 2, asparagine was dichotomized to low and high levels, and we saw again it was associated with CRC, showing that asparagine could be associated with poor overall survival and recurrence-free survival in female CRC patients. We then looked at ASNS expression using the Cancer Genome Atlas. And here we have Kaplan-Meier survival analysis, showing that high ASNS expression is associated with poorer overall survival in women with colorectal cancer. So here the red line is high ASNS expression, the blue line is low and medium ASNS expression, and you can see for females with both CRC and right-sided colorectal cancer that they have much poorer survival. But if we look at the males, we see there is no difference when you have high and low expression of ASNS. We then wanted to look a bit further at ASNS regulation. And then the literature shows that ASNS is an ATF4 target gene, which interestingly is upregulated under nutrient depleted conditions and regulated by mutant KRAS. We know that mutant KRAS is more frequent on the right side of the colon and in female patients. And we looked to see using TCGA data, if tumours from the right side of the colon that were mutant KRAS had different survival if you were female or male. And you can see again that for female RCC patients with a mutant KRAS profile that they had poorer survival. So our working hypothesis in summary is that asparagine production is increased in female patients with right-sided colon cancer and associates with poorer survival. Also, mutant KRAS and high ASNS expression associate with poorer survival for female colorectal cancer and right-sided colorectal cancer patients. So our current studies are really to examine the link between sex and ASNS regulation, and we're going to be using dietary studies and ASNS knockout studies in different models, and this work is funded by the American Cancer Society. And our aim is to design these precision medicines to target patient outcomes. But I think the take home here I wanted to put across is that tumor metabolism is different between males and females and associates with prognosis, so really needs to be studied further. But are these findings relevant to early age onset colorectal cancer? Well, we see that metabolomics is an effective tool for identifying these sex-specific metabolic differences in diverse populations and can be easily implemented into studies on early age onset colorectal cancer. Metabolomics as well, as I mentioned, could really reveal multiple influences that go beyond just genetics. So we can look at some of the environmental factors that may cause cancer or affect prognosis. So I'd just like to thank members of my lab and collaborators. With Dr. Xiaomei Ma at Yale School of Public Health, we're starting a study in early onset colorectal cancer using dried blood spots from birth cohorts in individuals that go on to develop early onset colorectal cancer. So we're interested to see this link between potential in utero exposures and early onset cancer. I'd also really like to thank American Cancer Society, NIH, and the Yale Cancer Center for funding my work.
Thank you, Caroline, for a fantastic presentation. And now we'll move on to Dr. Leung to round out our fantastic panel of speakers.
Hear me? Yeah. So my topic of today is using organoid models to study the biology of early onset colorectal cancer disparities. Okay. So this is my declaration of interest. And I'm a molecular pathologist and interested in the various pathways of colorectal cancer development. So this diagram summarized the two very important pathways, so-called the conventional pathway through the adenoma-carcinoma sequence and the serrated pathway of colorectal cancer development. And in each of this pathway, we have the majority going through the sporadic part. So the sporadic colorectal cancer through APC mutations and the sporadic serrated pathway through hyperplastic polyp that start with either BRAF or KRAS mutations. And then they all also have a familial part. The APC is well known. And recently in 2016, we described the germline RNF43 mutation as the familial pathway of the serrated pathway. And also we have the Lynch syndrome, which also go through two different paths. And with this kind of background, actually the serrated pathway is very interesting, in which it is characterized by early KRAS BRAF mutation, followed by either RNF43 or R-responding fusion, and then terminating in either the MSI or the MSS types of colorectal cancer. So this is histologically what the conventional adenoma looks like. We call it the progenitor phenotype that progressed to a colorectal cancer. And this is the so-called serrated pathway, in which we start off with a hyperplastic polyp that then gained either R-responding fusion or RNF43 mutations and progressed. Here is an example of where it progressed, with a, into a higher grade malignancy. So the interest in these two different pathways is intense because if the mutation resides on the upstream WIM upstream pathway, namely R-responding fusion RNF43 mutation, these are biomarkers used for clinical trial in which specific WIM inhibitor called a porcupine inhibitor that inhibits the secretion of WIM could be potentially effective in this group of tumor. And after our prior study for the interest in these two pathways, we move on to using the organoid model to try to build a collection of early onset colorectal cancer and look at their molecular features. And we note very interesting difference in age in relation to the key WIM pathway alterations. And with that observation from the organoid model, we go back to the TCGA colorectal cancer cohort. And this diagram summarizing the breakdown every 5 years with increasing age and the proportion of cancer that has either APC mutation or beta-catenin or R-responding fusion or RNF43 mutations. So one very obvious trend is the percentage of early onset of colorectal cancer with APC mutation is substantially lower in the early onset group. And so especially the younger you get. And you can see this branch that is in the, not related to APC, could be beta-catenin, and this is probably the Lynch group, and also RNF43 mutation and R-responding fusion, and also, none of the four. And so these groups actually is enriched in the early onset group and proven to be statistically significant. And then, the incidence of APC increase, and then you can see the drop at the extreme of age, beyond the age of 80 or 85. And then you start seeing RNF43 mutation more here. And this is the MLH1 methylation groups that, is enriching RNF43 mutation, in which we have also shown in our previous paper published in Gut. So with this background, we move on to, our organoid culture biobank. And the culture of intestinal stem cells, adult intestinal stem cell using this organoid model in Matrigel is discovered in 2009 by Sato and Hans Cleaver's group. So this is what it looks like. We can culture them in this 3D Matrigel, and they grow really in a 3D ball-like structure with a gland and a lumen, and you can image them with live cell imaging if you give them fluorescent marker. And this is, whilst there is mostly late onset colorectal cancer organoid biobank at the time when we published this paper. So this is the only biobank that is substantially enriched in early onset colorectal cancer. So this is the key driver genes that is observed in this biobank. This is the early onset group, and this is the late onset group. So if you look at the late onset group, they are, all virtually have APC mutation plus P53. And then if you look at the early onset group, there is this part that do not have APC mutation, but they are, either have R-responding fusion or RNF43 mutation, or in one of the case, that actually the mechanism underlying the auto-WIM signaling is unclear. And these actually have kind of a serrated phenotypes. So suggesting that there is, amongst the early onset group without APC mutations, R-responding fusion is one key group. And these fusion can be confirmed by RT-PCR. And, and this is the other case with RNF43 mutation. So we also have, our biobank also include a POE somatic mutation case. So somatic POE mutation has also been previously shown to be enriched in early onset colorectal cancer case in a study. And so interestingly, if you look at the POE mutation, it is only one hit, a single somatic mutation involving the hotspot domain of POE. And then you can see this trigger all these very high elevated mutation rates. And these mutations span, all sorts of driver like APC, P53, and HER pathway and PI3K, and etc. And you can see the very classical mutation spectrum of POE mutation. And this is also captured in the organoid culture. Interestingly, apart from this POE mutation, very high mutation rate, we also see another early onset colon cancer with a very unique mutation signature. And this patient actually is a leukemic survivor who has undergone bone marrow transplant at young age. And you can see this very heavy C2A mutation, and the mutation signature is not previously known. It doesn't match with any of the currently described mutational signature. And interestingly, this signature is not only seen in the tumor tissue, you can see it very clearly in the normal organoid tissue as well. And suggesting that there is a very big mutational insult in the normal colon of this patient that contribute to the cancer development. And we also search the TCGA tumor and see whether any of the tumor has this mutational signature. We managed to find one lung cancer patient that has very similar mutational signature. So this is not a very common mutation signature. So with the organoid culture, because it is nearly pure epithelial tumor with very few normal cell contamination, so we can get very good copy number profile of the organoid, and you can see the late onset and the early onset group, and the very characteristic of AP deletion and AQ gain in some of the both early or late onset and chromosome 20 gain, etc. So the organoid model actually quite closely recapitulate the in vivo situation. And interestingly, if you look at the mutant allelic fraction of the organoid and comparing the short-term and the long-term culture, you can see that this is, the mutant allelic fraction is maintained, suggesting that if there is a subclonal subpopulation, for example, with mutant allelic fraction of like 0.2, it is actually maintained. It maintained the heterogeneity, and this is the copy number, which is also maintained. So this compare the tumor organoid and its pair frozen tissue in terms of the mutation of these key drivers. So you can see that the organoid model actually closely recapitulate the in vivo tumor situation, suggesting that actually it is a very good model to look at, the tumor biology. Since the R-spondin fusion model has never been available before, so this is actually the first human R-spondin fusion colorectal cancer organoid model that we are aware of. The reason why this model is very rare and it's not represented in colorectal cancer cell line is because if you look at the growth factor requirement of these R-spondin fusion organoid, if you withdraw WIM, actually they all died. But they don't rely on R-spondin, so they are R-spondin independent. So given that all the cell culture medium is not enriched in WIM, so without WIM, actually you cannot grow this type of organoid. And explaining why the organoid model is good in a way it's very close to the in vivo situation. So we have also performed transcriptome analysis on these organoid, and we found that actually, interestingly, the R-spondin fusion organoid actually cluster with the normal organoid instead of the tumor. And the tumor here are mostly with APC mutation. And if you look at the WIM signaling pathway activation, so it is much more intense in the WIM signaling genes are the level is substantially higher in those with APC mutation compared with normal organoid as well as the R-responding fusion organoid, even though we are providing WHIM supplement in the culture medium. And interestingly, we also find a gene called PTK7 that is elevated mainly in the APC organoid, but not in the R-responding fusion organoid. And this gene is interesting because there are companies that are developing antibody drug conjugate to target this gene, and in an attempt to target the stem cell population. So you can see a differential application in these two molecular subgroups. We further confirmed the PTK7 elevated expression in the APC mutant organoid. You can see very strong fluorescent signal, but not in the R-responding fusion organoid. We also create a mutation, CRISPR in the mutation from a normal organoid, including creating a APC mutant organoid and an RNF43 mutant organoid. And you can see again, the PTK7 level is substantially higher in the APC mutant organoid. So we also perform single-cell analysis of these R-responding fusion organoid as well as the normal organoid, and look at the change in cellular dynamic. And this circus plot shows the cells that express a stem cell marker, transient amplifying cell marker like MYC, and also the androcyte marker denoted in green. So if we withdraw WHIM, you can see that the normal organoid all differentiate into androcyte with some secretory cells. So the stem cell compartment shrink. And this is what happened to the normal organoid. So in the two R-responding fusion organoid, interesting, you can see the behavior is very similar to the normal organoid. Without WHIM, the stem cell compartment substantially shrink, and especially in this case, they all differentiate into androcyte and some secretory cell. Whereas this is what happened with the APC mutant organoid. They don't rely on WHIM, and they still all have a very substantial stem cell population. We can also use lineage trajectory to look at the stem cell, early androcyte to late androcyte transition. You can see orderly maturation in the normal organoid, and also similarly in the R-responding fusion organoid. But in the APC mutant organoid, you can see they are trapped in a kind of a stem cell state. So we also look at the R-responding fusion organoid and look at what are the other associated molecular change. We see an enrichment of SMAD4 mutation in these R-responding fusion organoid. And then we look at another 3 database, because R-responding fusion is not very common. And you can see that in all 3 data sets, for those with R-responding fusion, there is a substantial marked enrichment in SMAD4 mutation in the organoid carrying R-responding fusion, suggesting that this combination of P53, R-responding fusion, BRAF, and SMAD4 seems to characterize the one very stereotype serrated pathway of cancer development. On the other hand, we also built an organoid model of the normal organoid culture, the normal colon, from these individuals. And interestingly, we see that in the normal organoid, actually, because individual crypt may acquire mutation throughout their life, and each crypt may have a different mutation compared with adjacent crypt, as demonstrated by Mike Stratton's group's work on dissecting the individual crypt for clonal sequencing. And so here you can see that the organoid culture actually capture these heterogeneous mutations. So their mutant allelic fraction is, concentrated around 0.1 or 0.2. But you can see that after long-term culture, they all raise to, as a population, raise to with mutation up to 0.5. So this is the river plot showing the clonal trajectory. So you can see that, this is the subpopulation that has a mutant allelic fraction of 0.2. After long-term culture, for most of them, they progress to 100% because of the mutant allelic fraction is 0.5 means that it's present in every cell in the organoid. And you also see that there are some driver mutation like ARID1A mutation in the normal organoid culture which get captured, and they may or may not get enriched, in long-term culture. So this is the interesting case that of the leukemic survivor with a lot of insult, a lot of mutation. You can see a lot of clonal evolution. Some clone disappear in long-term culture, and eventually one clone get dominant. So we have done it in like over 10 organoid, and they, if you culture them for 6 months, they all become clonal. So that means that there is competition between the normal cells, and eventually one of them would win out. So this is a more detailed analysis of the case with a lot of mutation in the colon. And you can see actually one hit P53 mutation, not uncommon. We saw 2 mutations here with a mutant allelic fraction of 0.15, but they don't seem to have a growth advantage in long-term culture. And you can see that, there is this shift, like in mutant allelic fraction from 0.2 to a clone that become dominant in long-term culture. So I think understanding of this is very important, because using normal organoid for study is increasingly used, like you can inject even the microbiome into the organoid culture. In Hans Cleaver's group have used it to show the Colibactin mutational signature. And so we have to understand a bit more about the biology of this normal organoid culture. And you can also CRISPR in mutation to create, combination mutations. And, you can actually create a cancer out of a normal organoid. And also, you can use it for large-scale drug screening. So this is a paper we published in 2018 using a gastric organoid biobank for large-scale drug screening. You can screen a large number of drugs, and here shows the very distinct response to NUTLI-3 for those with or without P53 mutation. So in conclusion, the organoid culture of early onset colon cancer capture diverse molecular repertoire that was not previously seen. And it also captured unique mutational signature and revealed combination driver mutation that characterize specific molecular subtypes. It can also capture subclonal architecture, and these remain stable in long-term culture in the tumor organoid. And you can do large-scale drug screening and also use it for study of cancer cell biology. And specifically here, the difference between the serrated pathway and the traditional adenoma carcinoma pathway, you can have therapeutic implication like oral Wnt secretion inhibitor, PTK7 targeted antibody, and these, is now in early clinical trial, and they may have potential in application between R-spawn infusion versus APC mutant colorectal cancer. And lastly, culture of normal colon organoid is a very useful resource for cell biology study, including use of CRISPR to recreate cancer through combination driver. And interestingly, these normal organoid culture, they capture the heterogeneous mutation that exists in individual colonic crypts, and then may exist subtle differences in growth characteristics of this individual clonal crypts, resulting in inevitable clonal dominance over long-term culture in vitro. And these growth dynamic is very interesting, and we wonder whether it may recapitulate the growth competition between several stem cells in a single colon crypt, that we know that it will generally overtaken by clonal dominance through neutral drift dynamic. And awareness of the existence of this clonal heterogeneity and eventual clonal dominance may have immense impact on study design using pooled normal organoids, so for biological studies and disease modeling. And also, these heterogeneity in normal organoid culture may be further explored for the study of preneoplastic events or even environmental mutagenic exposure, like the case that we show with in the leukemic survivor. So lastly, I would like to thank my group, especially Dr. Helen Yan, who co-lead the group with me, and a lot of members in the group and collaborators, both locally and overseas. Thank you.
Well, thank you so much, Dr. Leung, and I will go ahead and just round out the session with the path forward before we jump into our vibrant discussion based on all of the interesting results and information that y'all have shared. And I know Dr. Perea had to step away, and so this presentation is on both of our behalfs and really looking towards the future and how we can drive scientific breakthroughs to mitigate the disparities among patients diagnosed with early onset colorectal cancer. And so I've been so grateful that many folks have highlighted this figure over the last 2 days. This took a lot of careful thought and really putting together the different layers that comprise early onset colorectal cancer development and progression, and really highlights the complexity of studying early onset disparities and the different factors and different domains that are all interrelated that really yield early onset colorectal cancer development. And so circling back to when I presented that litter of 10 different facts that highlighted the disproportionate burden within this population, kind of would like to contextualize it in the context here, where you can see community health behaviors really fall into the built environment and have nicely shed light on the importance of examining community behaviors and social influences. Studies on both race and ethnicity in the absence of genetic ancestry is not available. Using population-based cohort studies as well as age-related analyses have highlighted the importance of some of the individual demographic factors, but also yield shed light on the importance of health behaviors and the impact that stress can have related to psychoneuroimmunology and driving colorectal cancer progression. And lastly, evidence that's emerging that leverages omics and biological tools that shed light on some of the molecular underpinnings underlying these disparities overall. But you can see that these are individual boxes. And when you kind of take apart these facts that I presented, it's challenging to really be able to cross some of these domains and really understand the interrelatedness and how these different factors may confound or impact and interact with each other to yield a distinct burden of early onset colorectal cancer. And so that kind of lends the question to itself, where do we go from here, given that much has been done, but there's so much left to do? And so kind of just capturing it in these different domains from the built environment, I think there is a need to further capture in more detail the social and environmental factor data from community health behaviors, where my team has leveraged publicly available American Community Survey data, et cetera, to things down to the family microbiome in the built environment that could be influencing some of these disparities that we're seeing present among patients diagnosed with early onset colorectal cancer. In the context of individual factors, there currently are colorectal cancer cohorts that collect data, but as discussed by some of the work that's ongoing by the early onset colorectal cancer work group, there's a need for a comprehensive individual level data collection and assessment, in particular the role of early life exposures, as Dr. Stoffel commented on antibiotic use and even antibiotic use in poultry or livestock, as well as better understanding genetic ancestry and not just using self-categorized or self-classified race and ethnicity will be critical. And on a biological and genetic perspective, both Drs. Johnson and Leung talked about leveraging different omic strategies or even combining omics together with mechanistic and lab-based approaches via patient-derived models that can shed unique light on the biology of early onset colorectal cancer and how this is really a heterogeneous disease within one population group. But really what the challenge is and what the need is at the same time is finding a way, like in that nature figure I presented, to really overlay each of these domains and be able to capture this data systematically for individuals in a large enough structure and with diverse enough populations so we can start to better answer some of these questions. And ultimately, it's the leveraging the integration of biological and laboratory-based mechanistic approaches with human studies to be able to use these patient-derived laboratory models and identify causal mechanisms that are driving some of these differences that may be yielded by changes in the built environment or together with individual factors, and could be mediating or driving early onset colorectal cancer development in different ways across population subgroups. And I know I speak for Jose here, where this isn't just a problem in the United States, as we've heard often throughout the last 2 days. I've gotten the comment previously that disparities are sometimes limited to race and ethnicity or in the United States. And I really hope that this session has really debunked that myth and highlighted that disparities persist across different domains, from individual to community level factors, from sex to genetic ancestry, the role of systemic racism, etc., and are really applicable worldwide down to geographic region. As you can slightly see illustrated here, that the proportion of individuals with early onset colorectal cancer sharply varies across countries, as Dr. Alatise commented on in Nigeria, and our studies have also shown that this burden sharply varies worldwide. And so this increasing incidence together with disproportionate burden suggests that we really need to tackle this on a global scale that will require close collaboration with diverse partners and sufficient resources to be able to do so in a comprehensive manner. And so to better address what biological mechanisms may be underpinning these distinct disease patterns, and really sheds light on coordinated efforts across countries, which is really the overall goal of this symposium, to begin addressing this important and timely topic. And overall, when it comes to early onset colorectal cancer disparities, the goal is to personalize prevention and detection strategies, as well as therapeutic modalities that can yield mitigation of this disproportionate burden, as well as the reduction of the overall global burden of this disease, that I think each of our speakers today have shed unique light on the possibilities where we can move forward and take some of this really exciting and promising work and better understand this diverse burden and this alarming epidemic across populations. And so with that, I will stop sharing my screen. And I know there's been some comments in the chat, but as the breakout sessions get moving forward, I want to encourage folks who are listening to please feel free to comment in the chat or the Q&A, and I'll also lead some discussion in some interesting and provocative areas brought up by our speakers. But I'll start with one question that will probably go to Dr. Stoffler-Johnson, related to what do we know about epigenetic factors and risk of early onset colorectal cancer and the role of the epigenome in addition to genetics. If you all want to take yourselves off mute, that would probably be a good way to get started to make this a lively discussion.
Yep, Dr. Johnson, I'm going to let you answer that one.
If you're familiar.
I wouldn't say I'm really expert on epigenetics, but we do see, in early onset colorectal cancer that probably the etiology lies with some sort of environmental exposure played out on a genetic background. So, environmental exposures do, affect the epigenome. So I think by having more studies in that area and pairing that, with some information on exposures or, other sort of life course history would be really important going forward. So,
Yeah, and I think piggybacking on that a little bit, the study that recently came out of UVA that showed differences in epigenetic aging of the normal colon between individuals who self-identified as non-Hispanic Black versus non-Hispanic whites tells us that even in normal tissue, there are differences, and they saw inverse associations and patterns between the epigenetic profile in tumor tissues versus paired normal that suggests there may be something attributable there that's to some of the early onset colorectal cancer disparities as well. And I think there was a follow-up comment that they related epigenetic factors potentially associated also with the microbiome, diet, and inflammation. And I think there's certainly limited data in this space, particularly for early onset colorectal cancer overall as a group. But that would be very interesting as data starts to emerge if we're able to further explore this. And I know Jose also commented on the study that he presented by Buchanan and colleagues regarding some additional age-related methylation drift. So not only is aging involved or aging-related drift, but now the complexity of potential differences in epigenetic profiles by race, I think also sheds light on the complexity again of this challenge. I think during Dr. Leung's presentation, we got the question of what proportion of early onset colorectal cancer is due to undiagnosed and or unrecognized serrated polyposis syndrome. And that's not something I am as familiar with, but perhaps Dr. Stoffel or Leung can shed a little light there.
Yeah, I don't know if it's been truly quantified, but just, from the bedside and from clinical perspectives of doing a lot of colonoscopies on young people, I've not seen a lot of serrated polyposis resulting in colon cancer in young people. In general, serrated polyposis we tend to see in individuals who are a little bit older, but I defer to Dr. Leung's experience.
Oh, you're muted, Dr. Leung.
So in the early onset colon cancer organoid cohort that we see, it's interesting that the case that has the R-responding fusion, for example, one of them actually have more than one serrated polyp in the colon in such a young age. So, but the number is not, high enough probably to be classified as serrated polyposis, but there's kind of seems to be like a field effect, a kind of trend, in this odd individual. And, whereas in the familial serrated polyposis, kind of a family that we have, described previously with RNF43 mutation, actually the age of onset is not that young, like in that family, the first patient that presents to us actually is, older than 60. But then when we trace the family member that has the mutation, actually some of them has a few hyperplastic polyps in their colon quite early. But I guess that, because these individuals would not undergo colonoscopy in normal circumstances, you may not have recognized them. So I think this area is still not very clear, and it's an area that deserves further study.
Yeah, thank you for that insight. And I think since we kind of touched upon the comment of colonoscopy, I kind of want to shift in that direction briefly because it's commonly noted in terms of sex disparities that colonoscopy is a more difficult or touted as a more difficult procedure in women compared with men due to differences in colonic length. And I'd love for Dr. Stoffel's kind of comments on that, and followed by Dr. Johnson's comments, because your paper spoke about potential changes in that gut milieu due to the change in colonic length that could be contributing to some of these sex disparities on a molecular level. So
yes, I can certainly speak to doing colonoscopies on young women. I take care of a large population of patients with Lynch syndrome and attenuated FAP. And I generally describe it as, the average colon is about 3 feet long. And if you are a 60-year-old man who is slightly overweight, the colon basically goes straight this way, and straight that way. Whereas if you are a, 5'4", 110-pound woman, you have about that much space, and you have 3 feet of colon in space about that big. So certainly, colonoscopy can be a little bit more challenging, but I think certainly in experienced hands, colonoscopy in young women and in children even is very safe and very doable. I think that one of the other questions that lends itself is that there's a lot of constipation in young women as well, because the colon is going not in a straight line, but making multiple loops, and whether that has an impact on the microbiome or potential toxic exposures in the colon to the mucosa is a big question.
Yeah, I think that's also interesting related to this. We see this proportional increased incidence of right-sided colon tumors among women. And so the potential attribution towards that as well, given that, of course, the colonoscopy is needed to detect the right-side colon versus the sigmoidoscopy. But Dr. Johnson, I would love your thoughts on this related to kind of what's known about the gut milieu with those changes in colon length, and kind of on a metabolic level, how that may be contributing as well.
Yeah, it's kind of interesting when you sort of bring the age component into this as well, and thinking about females and males, because obviously, when a woman is of younger age, she has a higher level of circulating estradiol, and there are some protective effects of that. So you have, activation of estrogen receptor, especially the beta isoform that's in the colon. But then that protection sort of lost somewhat after menopause, and they actually see, in later stage patients that there's more loss of this estrogen receptor beta isoform in the colon, and it tends to also predominate more on the right side, and that loss is more on the right side after menopause. So I think there's some interesting age differences that need to be looked into a little bit further. We had a study funded by NCI a few years ago, where we wanted to look at the differences in these bile acid metabolites that are in women and in men, but these were in older patients because we just didn't have enough samples, enough power to look at younger patients, unfortunately. We had about I think 12 people, so we did a really small study with the 12 people, but we did see that there were some differences in these genotoxic compounds and sort of the different types of bile acids that we found. And some of these bile acids are metabolized by the microbiome. So we saw these different distributions based on potential microbial activities. They sort of have to bring that into the context too, where are these microbes residing in the colon, and what are they doing? How are they metabolizing your normal dietary compounds, but also these other genotoxic compounds. And what we saw actually was that, so the females with right-sided colon cancer had these higher levels of these glycine adducts of the bile acids, and they weren't really being converted the same way that they were for the males. But then when we looked at the younger patients, actually the males with left-sided colon cancer had a higher abundance of these, where the females with right-sided had lower abundance. So even though we see these metabolic differences in older patients that seem to correspond by the region of the colon, I think this will be quite different for younger people, because they have different levels of these hormones that are regulating these receptors. They have multiple different signaling pathways involved in cancer. So it really is an area that needs to be studied more. From my perspective there, I haven't really been able to identify a lot of biosamples, to have enough power to do these sorts of studies, because we look at thousands of variables against a small number of samples. So, but yeah, I think that, colon length, especially when you do see these differences in microbial diversity across the colon and the types of metabolites that are being produced, which have beneficial and adverse effects on the microbiome and the biology, is really important. So.
Yeah, and can you speak to that a little bit with regards to the application of the metabolome? So I know we've actually discussed this previously before, and kind of the challenges that the tumor needs to be immediately frozen in liquid nitrogen to be able to timely and perfectly capture what's actually going on in a metabolic level, which may be a very challenging ask or something that may be difficult to kind of scale up with to be able to have some of these sufficient population subgroups. Can you talk a little bit about the application of the metabolome in the tumor versus previous studies? And in my experience, at least, we've done plasma metabolomics as well. So opportunities to leverage different biospecimen. Of course, they tell you something different, but how can potential, and you of course have targeted and untargeted metabolomics to complicate this even further. But can you talk a little bit about what each one may inform and how one may be a good start to be able to try and better understand some of these metabolic differences by sex or other population subgroups?
Yeah, so you have two types of way of doing metabolomics. You can do untargeted, which is like the screening approach. You look at absolutely everything that's present and it's unbiased in nature. But you do have the caveat that you're putting everything into the mass spectrometer or the NMR machine at the same time. So you get like things that are trying to be measured at the same time. So you get like augmentation and depression of certain signals. So it's not completely quantitative, but what it does do is allow you to identify metabolites that you may never have thought to be associated with your phenotype. So it's completely not hypothesis driven, and it was never really appreciated for a long time. But I think now that we've found some really novel things, it's now becoming more common. For targeted metabolomics, you have a set of standards that you've already run on your instrument. There may be like 100 metabolites or 500 metabolites, but what they are, where they reside in biology, metabolic pathways, and you can sort of try and piece together what's happening by using that sort of instrumentation. So I guess it just depends on, the bandwidth of the group and, what your study is about. But in terms of sample type, looking at blood and urine is a great way to sort of potentially develop a biomarker because it's non-invasive. So if you do find something, then you might be able to go back in and screen everybody, so if you find a biomarker for stage 1 right-sided cancer in the blood, that would be amazing. You could screen everybody for this early onset, early stage cancers, but they just haven't really been able to be replicated. So there's been lots of studies that have been carried out. There's only one real biomarker that's come from metabolomics work so far, and that's for cardiovascular risk, and that's sort of come out of Stanley Hazen's group, the TMAO. So I mean, my personal thought, this is just my thought, is that going to the tumor itself will give you understanding of the biology of the tumor. If you go to the blood or the urine, it's really confounded by things that are in the diet. So there might be very small signals that are really important in the tumor that are just lost because of a dilution effect, basically. So we try to go from tumor source and then out to validate in blood and urine. But it's just having those biobanks and the accessibility of those tumors, which is difficult. So
Absolutely, and appreciable for the work that you've been able to do with that, with those challenges and the approach. I think one of the interesting things you touched on a little bit, and kind of taking this into the laboratory setting with the organoid, is that the organoids have been, as elegantly shown by Dr. Leung and her group, that the organoids are able to recapitulate the genomic profile of the in vivo frozen tumor tissue. And Dr. Leung, you briefly spoke a little bit about the potential to investigate the microbiome in organoid culture. I know in your study you conducted different omics single-cell sequencing, leveraging this novel model system. Can you discuss a little bit about the potential to also investigate some of the other omics, such as the metabolome or potentially even the proteome, that could shed additional light on a multi-omic scale? I'm a little biased. My work is predominantly integrative with multiple omics at the same time. And so naturally I kind of jumped to, oh, well, can we interrogate the metabolome or the proteome and shed some additional light in that direction?
Certainly, you can grow those organoids and extract enough protein for untargeted proteomic profiling. And I think some other group has published paper on that. And for metabolic profiling, I suppose it's still okay. I'm not very familiar with the area. I think the only limitation for the organoid is, they don't grow as fast as cell-lined, and they are more expensive to grow, compared to cell line. So, if you need a lot of cells to perform your profiling, then it's become challenging. But if your requirement of cell number is not as great, I think it's quite achievable with the organoid culture. Yeah.
And I think you also nicely shown the potential to leverage CRISPR knockout systems in the mutant organoids. So it really kind of connects again from the human study down to the laboratory-based mechanistic perspective, that potential identification of a novel gene associated with early onset colorectal cancer on a somatic level, and potentially be able to understand if you perturbed or knock out that gene in the organoid, the downstream consequences and the pathways that may be affected to further understand the actual mechanistic underpinnings of the disease, and also be able to discern if you were able to culture organoids for men and women or sufficiently across some of these diverse population subgroups and knocked out a gene of interest. Do you see the same downstream consequential effects, or are there potential changes by sex or across racial, ethnic, or genetic ancestry subgroups overall as well? So I think kind of seeing that potential and leveraging that model system harbors a great deal of power as well as skill to be able to culture those organoids and do so successfully. So kind of seeing your group lead the way and demonstrating that it can be done, and how well it recapitulates the patient's tumor for early onset colorectal cancer, I think is a really promising and direction and approach overall. There was an additional question that asked to clarify related to your work, that it seems like serrated pathways are playing a relevant role in a proportion of early onset colorectal cancer related to the study in Gut U Illustrated. Is that a correct statement, or can you further elaborate on that a little bit?
Yeah, I think we do see, although our cohort is too small, right, to reach statistical significance, and we also try to look at the TCGA data and see whether statistically we can prove that early onset colorectal cancer is enriched in, for example, R-responding fusion. I think it can't reach statistical significance, but given if you see that there are less APC mutation in the younger age group, which is very clear cut, right? It has to be something if it's not APC, right? So I think it's the constellation of a mixture of like R-responding fusion or the Lynch syndrome case with beta-catenin and so on that constitute the rest of the case, without APC mutation. And I think the interesting thing about the R-responding fusion is actually a chromosomal break and, translocation, right? So that types of change probably, may happen in the earlier age, because, if you think about all the leukemia, for example, a lot of them is due to translocation that induce a stem cell pathway. So I think this is probably a very different mechanism, compared with the traditional APC mutation, which seems to be very age-related APC. The older you are, the more likely you get, an APC mutation or some APC mutated crypts in your colon probably, right? Yeah.
Elena's nodding her head, yes.
Yeah, no, I think that's absolutely right. And that's what we see is that, if you look at your garden variety older onset colorectal cancer, 80% of them will have somatic APC mutations. If you look at colon cancers in young individuals, you don't see that same proportion of APC mutations. And likewise, when we look at young colon cancers, we see that the vast majority of them are microsatellite stable. The ones that are not microsatellite stable, the ones that have MSI high, are almost all Lynch. Not all, but almost all Lynch. So we know that there's something else going in there that is going through the microsatellite, not through the microsatellite instability pathway. And I guess that's why, certainly there are some young onset colorectal cancers that are going through the serrated pathway, but I don't think that explains the majority of what we're seeing.
Yeah, and so touching on that point related to the germline genetics, both yours and Heather's seminal papers came out near the same time to demonstrate that 16 to 20% of early onset cases harbor a germline mutation in a known cancer susceptibility gene. However, to my knowledge, not much whole exome sequencing of the germline has been done. Is there potential that there may be some novel pathogenic germline mutations that could be associated specifically with early onset colorectal cancer? Or I'd welcome your thoughts on this a little bit.
Yeah, so Laura Valle is also in the audience, and so she can certainly weigh in because she has done a lot of the work looking at comprehensive germline sequencing in young onset colorectal cancer patients who do not have identifiable pathogenic variants in any of the known suspects. And, through that comprehensive sequencing, we have found things like POL-E and POL-D1 and RNF43, but these seem to be kind of rare and few and far between. I think that the majority of the sequencing work that has done looking beyond the usual suspects at whole exome and whole genome sequencing have not identified a smoking gun.
Yeah, and absolutely. And I think, albeit always harbors down to sample size, I think it would be very interesting to take a look at the prevalence of some of these mutations by race and ethnicity, which seems to potentially suggest there's some interest there. I have some ongoing work examining some data that potentially, suggests that these patterns will differ by race and ethnicity and warrant some further study. But again, of course, it's very challenging. I think as each one of the panelists has commented, that not only are you looking at a small proportion of all of the colorectal cancer cases seen, but then to try and be able to have sufficient power to look within some of these different subgroups while accounting for meaningful differences by stage and site. And that adds another layer of complexity to this pool and really drives the emphasis of needing large-scale collaborations to be able to tackle some of these questions overall. What struck me, Lena, in your presentation was the comment of the proportion of cases with no pathogenic germline variant was higher among rectal cancer patients, I believe, if I recall correctly. Can you speak a little bit further on that point?
Certainly, and that's what we're seeing epidemiologically, right? So the largest rise in incident cases is among rectal cancers, and the rectal cancers overall are less likely to be associated with pathogenic germline variants. And so that leads us to believe that there is kind of an environmental exposure component to what we're seeing that is not necessarily associated with a strongly penetrant germline predisposition, but rather that points in the environmental direction.
Yeah, absolutely. And I kind of want to shift over to a little bit on the racial and ethnic disparities topic, because I think as evidence is beginning to accumulate both epidemiologically and biologically could suggest some meaningful differences. You highlighted our study where non-Hispanic Black individuals diagnosed with a stage 2 colorectal tumor had a 60% increased hazard of death. And we posited potential differences in cancer treatment or potential differences in tumor biology. Can you speak to that a little further, kind of that hypothesis? And I know work is very limited in this space, and we recently showed that non-Hispanic Black individuals using the AACR Project Genie database carry a higher tumor mutation burden, specifically among early onset cases compared with whites. It persisted for both early and late onset, but it was a striking finding given the potential therapeutic implications of this difference. But I'd love to hear more a little bit about that, because it was always striking from the moment we found it that the largest disparities were not among cases with advanced stage 4 disease, but the disparities that we observed that were most striking that did not persist between Hispanics and whites were in that stage 2, stage 3 colon and rectal tumor group.
And you're exactly right. And thank you so much for calling attention to that, because I think that what we know about stage 2 colorectal cancers in young people is that this is kind of a little bit of the Wild West. Because if you look at the data that we have from the behavior of stage 2 colorectal cancers in older individuals, it basically tells you, number one, that adjuvant chemotherapy doesn't make a huge difference in survival. And number two, that people tend to do pretty well. But what we do know with some reports of young onset colorectal cancers is that even stage 2 tumors in young people are more likely to have poor prognostic features. So they're more likely to have tumor budding or they're more likely to have tumor budding and lymphovascular invasion. And so what we postulated on the basis of the results saying that the biggest differences we saw in outcomes were among individuals with stage 2 disease was that perhaps the individuals with stage 2 disease who were treated with adjuvant therapy did better than the people who were not treated. And one of the limitations of our study was that we did not have treatment data. But I think that what we were looking at was saying, well, perhaps there was a difference in how the individuals with stage 2 disease were treated in these two populations based on your race, ethnicity, and where you lived.
Yeah, in the adjuvant setting, yeah, we of course had at that surgery and radiation, but in terms of chemotherapy, that is not available in SEER and is largely incomplete. But yeah, I think that calls upon these multiple domains and layers, from the quality and access to care and the potential that has, in addition to the role that genetic ancestry or sex plays into the development and disease progression of early onset colorectal cancer in particular. You raise an interesting point in terms of some of the pathological features like tumor budding that has been gaining clinical relevance overall. And so it's kind of tying it back to the organoids, because one of the really unique and interesting features of leveraging an organoid model versus a 2D cell culture model is the ability to kind of recapitulate that 3D environment. So Dr. Leung, can you speak to the potential, although it's still an artificial environment and requires some of the different growth factors in media to culture and populate, do organoid models harbor the potential to be able to look at some of these features such as tumor budding and be able to assess that given their 3D nature?
I think it's, we can assess the growth rate, and also, in the organoid model, they also kind of have a different shape, like sometimes they are more solid, if they are poorly differentiated, or, they are more cyst-like if they're slow growing with a lot of budding. But I'm not sure whether it can totally recapitulate what the tumor budding in vivo, because I think that probably also related to a tumor stroma interaction, how they invade or, grow. So there are, attempts to engineer environment to put in the stromal cells and, using the tissue engineering kind of approach. So I think maybe when that type of model get better, build, then you may be able to see more and more this, assay to reflect that types of activities.
Yeah, absolutely. And I think it's a little ambitious, even when I just said it out loud, because tumor buds are, of course, such small clusters of, 1 to 3 cells that it's a lot easier to assess in an H&E stained slide and is even subjective to some extent. But I think just highlighting the advantage that leveraging, the patient-derived organoid system has with recapitulating a little bit more of that environment and that 3D nature that cell lines are unable to capture in that 2D setting is, I think, a really key point. Despite the slower growth time and the higher cost of that model system, I think there are really key advantages of kind of positing to leverage this moving forward in diverse patient populations overall.
I just wanted to also weigh in that I use tumor budding as an example because it has been described in the literature and has been described primarily in the older onset colon cancer literature, but it is possible that there are other histopathological or molecular features in young tumors that may actually have better prediction for outcomes and that we just haven't even gotten there yet.
Absolutely. And I think that speaks to also the level of individual level data that's truly needed. While population-based cancer registries and large epidemiologic cohorts have their advantages, some of these, and I speak from both an early onset colorectal cancer as well as an early onset appendix cancer perspective, some of these features that are really critical in prognostic value in particular are pretty challenging to count. As you grow in scale of the data, you lose some of that granularity, and investigating some of these features on either a single institution, which has its challenges and limitations, or having that granularity of data, I think, is going to be also really important moving forward, to not just to understand that biology of the tumor, in addition to some of the germline components that could be weighing in, or how the tumor is able to evolve or evade. Of course, there's lots of discussions around chronic stress and the role of systemic racism contributing to stress and that potentially influencing colorectal cancer progression, which I think is also a topic in and of itself overall. But I think
it's a potential issue of sleep.
Absolutely. Stress and sleep connected to diet and physical activity. It all requires a very, very detailed capture of data, which can certainly be challenging. Where you go in depth in one area, you may lose some of that granularity in another.
Sorry, I think not to harp on about environmental exposures, but I think they do really affect too your immune responses. So I don't really think it's been evaluated to any depth, like for people that have cancer, what effect, like where you live, air pollution, what is in your drinking water, how does this affect cancer progression? How does it affect your immune responses and respond to treatments? And that there are major disparities as well in that area. So just wanted to add that point.
That's a great point. I think that speaks to community health. And from my group's experience, we've tried to use some of that community health data in looking at county level physical activity for adults or adult proportions of obesity. But then, of course, you're kind of grouping a region together where there may be heterogeneity within this group. And at the same time, I think the importance of residential history is now growing as the early life exposure component is growing, because some of these exposures, if someone reports residing in a certain area, how long have they resided there? Had they had a previous exposure we may not capture? And I think that speaks to the complexity of the human overall, and the different factors to consider that complicate this, but also make it a really important area of assessment to better understand so that we can mitigate some of these really critical disparities that are emerging within this population, particularly as, Elena said, most of these cases are microsatellite stable or sporadic, suggesting that these individuals might be at average risk and may not be eligible for any screening until age 45. And so in that event, while lowering the screening age has been widely discussed and celebrated as a fantastic win that 45 is the new 50, there's still that need to identify who may be at higher risk for that microsatellite stable tumor overall. But I think kind of tying it back to the clinic and overall to what's been discussed, just recently I had a couple interactions with patients where, Elena, you commented on reaching that primary care provider with respect to collecting family history of cancer, and how to do that, particularly in the context of the setting in the residential area, even perhaps. But sometimes even the family history of cancer can be a taboo topic for patients, and kind of even being able to understand it from their family members, or yesterday in the patient's story, after a diagnosis for one of the patients, then they discovered that familial history of disease. Can you comment some of your thoughts in the context of that component here, and just kind of better characterizing and understanding this, especially in populations with this disproportionate disease burden?
Yeah, absolutely. And I think that we are fortunate to have received a UO1, one of the NCI UO1 Cancer Moonshot grants, looking at how to expand access to cancer genetic testing to cancer patients throughout our state in Michigan. And the first aim of that study is actually to introduce a family history ascertainment component, and we're working with both oncology practices, 68 oncology practices in the state, as well as working with patients and families to raise awareness of the importance of taking a comprehensive family history, one that doesn't just include colon cancer in the family, but actually all types of cancer, to be able to assess genetic risk. And part of what we're doing includes outreach to the Middle Eastern, North African communities, where talk of cancer is often taboo, and to provide an opportunity to educate about the importance of discussing cancer diagnoses, because it makes a difference for families. And I think that a lot of this is working with patients and communities and providers to provide that level of education and that investment and that self-efficacy. So we're very excited about this group work. Yeah,
that's very, and I mean, I maybe will just chime in one thing, because I, of course, trained in Detroit. That Dearborn in Michigan is actually home to the largest Middle Eastern North African population outside of the Middle East. So I think there's a real marked opportunity to kind of fill that gap and make a huge difference for that population in particular. So I will certainly look forward to hearing your findings and discerning kind of where we can shift the needle to make those discussions a bit more dinner table friendly or common or less taboo overall. And I think in the final 2 minutes of this fantastic and engaging breakout session, I'll kind of just leave it to each of you if you have any closing remarks you'd like to make on this topic of early onset colorectal cancer disparities, and really kind of where things are going and headed. And of course, I want to express my absolute thanks to each of you for agreeing to serve on this panel and give fantastic presentations that I think have really spanned from cells to society, or showing that society meets biology, and that this is a complex interplay of factors that we need to disentangle. And so I'll leave closing remarks to each of you, and also thank everybody for attending the session as well as staying on for the breakout.
you so much for inviting us. And I think that one of the wonderful things about this symposium is the multidisciplinary nature. We have patients, we have scientists, we have clinicians, and I think everybody needs to contribute to this conversation because we all learn so much from each other.
Thank you for inviting me to join this. So although a lot of the disparity is talking about, the population that does not seem to include the Asian population, but I think, there is very interesting difference between the Caucasian and the Asian population as well. So I think, collaboration in that area would be also very interesting. Yeah.
Yeah, certainly. And I think I can speak for myself and Elena. When that JCO paper came out, we actually did not have sufficient data for the Asian or Pacific Islander population across the United States to be able to include that important racial and ethnic subgroup. So I think that's also a true area that we can really better understand, not just across racial ethnic subgroups, but the population of Asian and Pacific Islanders is also broad and heterogeneous. And so also understanding some of those differences as well.
I just want to thank you for inviting me to talk. I think it's a really great session, Adriana, and a really important topic. So thank you for highlighting, the disparities in this area. And I look forward to hearing more and being involved in early onset colorectal cancer more.
Yeah, well, thank you to, again, the speakers and everyone for attending this perfectly. We're right on the dot, rounds out our time. And I'm sure that both myself and the speakers are available for any additional questions offline. And this remains an ongoing discussion. And this is not something we'll be able to mitigate immediately, but I think the important attention and efforts in this direction will help us make paradigm shifting breakthroughs for what's most important, and that's the patients. And so again, thank you so much to everyone, and have a fantastic weekend because it's Friday afternoon here. So I hope everybody enjoys their weekend, and thank you again to everyone for attending.
Thank you.